Cool. Hi everyone, I'm Maria. I'm the Events Officer for OUCB, and just introducing today's talk, a big thank you to Professor Fraser Armstrong for coming, and he's going to be talking to us about the importance of enzymes, electrons, and nanospace for the future of catalysts. So handing it over to you, Fraser. Thanks so much.
Thank you very much, Maria, and thank you for the invitation to speak and present some of the work that my group has been doing for probably 20 to 30 years. So from the title, you can see that we're going to be talking about electrocatalysts. And that's really the first part of my lecture. It's just going over a few aspects of why enzymes are the world's greatest electrocatalysts, which is something that I don't suppose most people appreciate. But to understand why enzymes should be so good at catalyzing the interconversion between chemicals and electrons, we need look no further than the natural world. Because if we take as one example, the hydrogen cycle, hydrogen is used by microorganisms as an energy vector. It's used to store energy, and it's used to accept electrons in a rather crude respiratory chain, where the proton is the only available oxidant in the system. And the hydrogen cycle is exemplified by bacteria and archaea. In the atmosphere, we have about half a ppm of hydrogen, and a whole host of organisms can make a living out of this very low hydrogen level. This is an old slide from an article I wrote about 15 years ago now, or nearly 15 years ago, with Kylie Vincent and Alison Parkin, when we talked about the use of the electrochemical methods to look at hydrogen. We can consider that electrocatalysts would have evolved at that time when primitive life was evolving. And the evolution of electrocatalysts is an issue that won't be familiar to most industrial people, because they would think that electrocatalysts started with William Grove, but in actual fact they probably started well over 2 billion years ago and have evolved to some perfection ever since. What I'm going to show here is an example of a respiratory chain. So this is what happens in our mitochondria. And it's like a spectrum. As you read from the left-hand side to the right-hand side, what we're doing is we're taking electrons from a region where they have a high energy in substances which we recognize as fuels, that'd be sugars and hydrocarbons, to a potential region where they have very low energy, and the electrons finally end up being donated to oxygen, and the net result is energy which is released for use by the organism. And each of these lines, particularly in the green zone, represents a metabolite, and the potential difference between these different metabolites is often very small. So in other words, for hydrogen to be oxidized by NAD, one often only has a potential difference of tens of millivolts, and yet these reactions occur very rapidly. So we're thinking here about the efficiency that living organisms must have developed in their catalysts to carry out these reactions. In the next slide, what I'm showing here is a typical enzyme that is responsible for producing energy. This is a hydrogenase, and this particular one comes from gut bacteria, the common E. coli, and on the left-hand side, we see it's seated on a membrane. It's seated on the cytoplasmic membrane, and this enzyme catalyzes the interconversion between hydrogen and protons at a buried center in either side of this heterodimeric structure. So it's a dimer of dimers, the catalytic entity of the enzyme, and it sits on these anchors here. In such a way, we can envisage the hydrogenase being seated on an electrode surface, so that instead of the electrons flowing eventually into the quinone pool located inside the membrane, the electrons flow instead into an electrode. And of course, by reversing the potential on our electrode, we should be able to make electrons move backwards to convert protons into hydrogen. Now, these are large molecules. This heterodimer, a dimer of heterodimers with other ancillary proteins, has a molecular weight of well over 200,000. And so it would be at first surprising to find that it's such a good electrocatalyst. This slide here depicts the concept by showing on the left-hand side a cartoon. So this is an enzyme. We have a buried active site, as we typically do. The active sites are often shielded from most of the solvent. And the active site, which is catalyzing the transformation between reduced and oxidized forms of a molecule, has to have a supply of protons, and which can come into the active site through various pathways, and electrons. And electron transfer is easy to see in an enzyme because it occurs on very noticeable transition metal relays. In this particular case, these cubes represent iron sulfur clusters. This distance between the active site and the exit point on the enzyme may well be over 50 angstroms. Now, on the right-hand side, what I show are two extremes of the readout of the experiment that we're going to be talking about for the next 40 minutes. This is a voltammogram, and it's a voltammogram that is obtained when I have an electrocatalyst, like an enzyme, seated on the electrode, and it's catalyzing the interconversion of O and RH2, which are present as a 50-50 mixture in the solution. Now, the blue voltammogram that you see here is actually rather special. The point where I indicate here, where the current axis, which is the vertical axis, and this crosses the electrode potential axis, this point is the point at which the two sides of that equation at the top are in balance, and the rate is occurring at an equal speed in either direction. And if I nudge the potential 1 millivolt to the left or 1 millivolt to the right, the direction, that is the net direction of the reaction, changes. We call this a reversible electrocatalytic voltammogram. It is very unusual, and the only common example in industry is the platinum electrode, which is reversible for the hydrogen proton interconversion, and which we can use to power to energize a car, a hydrogen powered car. It is very rare because most electrocatalysis, in fact all of it outside the platinum electrode and biology, exhibits a curve which is given by this red line here. And what we can see here is that if I apply an oxidizing potential, so I'm now trying to drive that reaction RH2 over to O, there is quite a region of potential where nothing is happening. And then at a certain potential here, the reaction starts to go. In various ways, I would refer to this difference between the reversible case and the normal irreversible case by this eta symbol, which means overpotential. So an overpotential is required in almost all cases to get a reaction even just to occur, let alone reach a reasonable value. And I think one can see here that if I took this value eta and if it was a value of 100 or 200 millivolts, which is actually small by many industrial standards, life would not exist, because the mitochondrial and other respiratory chains that I showed would never occur at a sufficient rate to make them viable. So I'll come back to that in a short while, but the experiments that we do are actually very simple in essence. We take a pure sample of an enzyme, and we've studied many different enzymes over the years, and we would usually use a small electrode that can be rotated at high speed. The reason for this is that rotating the electrode in a solution creates a hydrodynamic flux, and so the electrocatalyst on the electrode surface shouldn't deplete the reactant that is coming up towards it. There's a continuous supply of reactant. And in a typical experiment, we would take a very small amount of enzyme and allow it to absorb onto the electrode surface. We find that enzymes are generally very sticky when it comes to binding on surfaces. And a favorite surface of ours over several years has been pyrolytic graphite, which is, you can see here, if you're familiar with graphite, you would see that all these lines are represent the layers between the graphene rings. This is actually quite a crude electron microscopy experiment here, but you cut that with a knife. You cut it so it's perpendicular to this, and you expose the ends of graphene rings, a huge number of them, which makes a hydrophilic surface. What you can see on this is that the surface hasn't been sonicated, so there's still fragments of alumina which are still on that surface, which we would usually get rid of. Now, I'm just going to return to the experiment, because as I pointed out, the voltammogram on the left is very special, because this little green oval here is representing the reversible region. And the voltammogram on the right is a less fortunate electrocatalyst. It needs quite a large overpotential to get it to work. Over the years, there have been many articles which deal with the efficiency of redox enzymes. Some of them originated from Oxford back in the 1970s by Jeremy Knowles and John Albury. And the emphasis all the time is on turnover rate, how fast an enzyme works. It's the vertical ruler here. And the best enzymes would work at a diffusion-controlled rate. This would be the normal method of, or the criteria for talking about efficiency. When we are dealing with enzymes as electrocatalysts, we have another variable, which is the potential that you need, it's the horizontal ruler, to get the reaction to occur. So, I'm going to give you now quite rapidly some examples of enzymes that we've studied and which display reversible electrocatalysis. The first one, this is a hydrogenase. It's a nickel iron hydrogenase, I show the active site on the right. There's an iron atom, which is it's like the sort of complex that you see in organometallic chemistry with two cyanides as a CO, and that's coordinated by bridging cysteine sulfurs to a nickel, which also has another couple of sulfurs. Well, this strange active site is a reversible electrocatalyst for the hydrogen reaction. And you can see that here, the current cuts sharply through the zero current axis and switches direction at that point here. Another example, this is not one from my lab, it's from Judy Hurst's lab in Cambridge. This is formate dehydrogenase. And of course, now we're dealing with carbon chemistry, the fixation of CO2 in particular. And this has a molybdenum atom at its active site. And again, you can see the telltale quality of reversibility. Formate oxidation and CO2 reduction are connected by simply a few millivolts or so in potential. This is obviously an extremely efficient enzyme. This is a different type of enzyme because this one reduces oxygen to water. And what I show here is the electrocatalytic voltammetry of an enzyme called bilirubin oxidase. So that is reducing oxygen. The green marker here represents the formal potential of the 4-electron oxygen to water couple at pH 5. What you will see is that this enzyme is not acting reversibly. In fact, there is over 100 millivolts of overpotential to be supplied before oxygen reduction starts working. But it is still, at pH 5, considerably better than a platinum electrode, which you see on the left-hand side here. Now, the active site of these enzymes, they're known as blue copper oxidases, consists of an active site of 3 copper atoms. And this is held in the interior of the enzyme, and a short distance away, I say short, it's about 10 angstroms away, on the surface of the enzyme is an electron relay site called a blue copper center. Now, several years ago, we proposed that a major contributor to electrocatalytic reversibility was that the reduction potential at which an electron enters or leaves the enzyme should be roughly commensurate with the reduction potential of the reaction being catalyzed. Otherwise, the bias of the catalyst will be too far in either direction. So this experiment was carried out at pH 5. And as all of you will know, certainly if you've done chemistry at Oxford, you will know that if you have a 4-electron, 4-proton reaction, then its potential will get more negative by 59 millivolts for every pH unit. Now, the point here is that the oxygen to water potential does drop by 59 millivolts per decade, but the potential of the copper site, the blue copper site here, which is the electron transfer site, does not. It's much more invariant with pH. And so a few years ago, we proposed that if we took the pH up to 10.2, in fact, we were very precise because we could predict a sweet spot at which this enzyme would now become reversible for the 4-electron oxygen to water reaction. And we were right. The electrocatalysis is very feeble, actually, and it's not very stable. In fact, the cyclic voltammograms that you see here decay with time quite rapidly, but they carve out isosbestic points. And the isosbestic point is exactly as you expect for the oxygen to water potential. So we have an example here of an electrocatalytic concept being proven, although the use of this is really close to zero. This is another enzyme that my group studied. This was first looked at by Alison Parkin in 2007 and then followed up later by Vincent Wang. And this is the carbon monoxide dehydrogenase. This enzyme actually occurs in by the sides of volcanoes, and it occurs in bacteria that live on in the chimney flues. And this organism, which is responsible for producing this enzyme, note it's got a nickel as part of an iron sulfur cluster here. This enzyme can live on carbon monoxide. Well, look at the electrocatalytic voltammetry. Here you've got the mark of reversibility. CO2 reduction to the left, CO oxidation to the right. And this particular reaction has been so inspiring to physical material electrocatalyst designers that it features as a benchmark in a paper several years ago by Jens Nerskov. Jens Nerskov is one of the pioneers of electrocatalysis in the energy sciences. And what he showed as one of his figures was the electrocatalysis of CO2 to CO interconversion as it occurs at a gold 211 surface that he'd carefully prepared. And the voltammetry looks like this. You can see how irreversible it is. And what Nerskov had put onto the plot was an overlay of the result that we'd obtained before that with carbon monoxide dehydrogenase. This is the reversible electrocatalysis. This is the best, or one of the best, that industry can come up with. Now, in the next part of my talk, I'm going to just discuss an experiment that was carried out 2 years ago in my lab, when a student from Thomas Happer's group in Bochum in Germany came over to spend a few months working on an iron-iron hydrogenase. Now, this is a little bit different to the nickel-iron hydrogenase. It's rather more complex. You've got 2 ions, you've got cyanides and carbon monoxides. You've got a strange bridging ligand. It's coordinated to these 2 ions by sulfides here, these thiolates, and at the top of the bridge here is an amine. This is a secondary amine, and this secondary amine looks over the distal iron atom. I call it distal, proximal, and this is the other part of the H cluster. It's an iron sulfur cluster that serves as an electron buffer. Well, what happens here is that hydrogen binds to this just slightly electron deficient iron 2 site here, and it's cleaved heterolytically by a frustrated Lewis pair mechanism, which leaves a proton on the nitrogen and a hydride on the iron. And the electrons associated with this iron 2 hydride are eventually dispersed out of the enzyme via this 4 iron 4 sulfur cluster, and they go towards the left as I show it on this picture. So this is the H cluster, the hydrogen activating active site of the hydrogenase, which occurs in an organism called Clostridium pasteurianum. It's a strict anaerobe. Well, what Oliver Lamprecht looked at was a set of variants of the iron-iron hydrogenase in which proton transfer sites had been modified by genetic engineering. So two groups on a proton transfer relay pathway, which had been established just about the year before, were found to show just slightly slower rates of hydrogen activation. And the two residues, there's a glutamate here, glutamate 282, which was converted into another residue, and also another glutamate here, glutamate 279, which was also converted into another residue. Now, the other residue has just one CH2 group missing from the linker, which links the carboxylate side chain to the main group. And so it's slightly short. And this effect, this slight variation, either mutation 2 or mutation 4, had a very interesting effect on the catalytic voltammetry. Now, what I'm showing you here are some examples of when we've mutated either of those two proton transferring residues in Clostridium pasteurianum hydrogenase 1 or another hydrogenase. And you have to remember here that the protons are coming to the H cluster active site in one direction, and the electrons are coming to the active site in the opposite direction. And as electrochemists, we're picking up the electrons, but we can only see the electrons if they've been produced by catalysis or consumed by catalysis. So I hope you can see these here. What I've got are some black lines which cut sharply through the potential axis. Let's just focus on the top voltammograms. These experiments at the bottom were carried out with another iron hydrogenase, which comes from photosynthetic algae. But just look at the top ones. What you see is that the sharp cut through has been replaced by an inflection. You see this inflection appearing? It's very prominent in some of these cases. Well, this inflection is the emergence of irreversibility. It is a very mild effect. It could not be detected by any other means, but it is unmistakably appearing in the electrocatalytic voltammograms. And the explanation for this is that the electron and the proton motions, which are coming in different sides to different sides of this macromolecule, must occur in concert for the reversible electrocatalysis to take place. I indicate this with through two diagrams. These are the same diagram drawn slightly differently. The production of hydrogen from protons requires would require two electrons and, of course, two protons. And it resort here to a very important concept in electrochemistry, which is called the square scheme analysis. These are effectively thermodynamic cycles, and the most oxidized form of the active site is linked to the most reduced form of the active site by two electrons and two protonation steps. Now, I've turned this round here. You see, you can either move your proton first, or your electron first, or your proton first, or your proton first, or your electron first. In either case, or you can do them both at the same time. So turn this diagram up on its end by 45 degrees, and what we see here is a distinction between the reversible case, which can occur when the electron and the proton move in concert, and the stepwise case where the proton can't move at the same speed as the electron, and so you move an electron first, but of course it needs a little bit more potential to drive that electron on its own. When it's assisted by a proton, the charges compensate each other. This is another way of looking at it. And so under oxidation conditions, if I don't have an extremely efficient proton transfer pathway, then the system is going to tend to move towards the right, putting an electron on at a bit of extra cost, then allowing it to relax back with a proton, move another electron on, and then it allow the system to relax back with the proton, all of which is much slower than if I could just do them all at the same time. Likewise, in the reducing direction, we have to move an electron first because the proton is too slow, but then we drive the proton on, and so on. And so the point here is that proton-coupled electron transfer is a very important factor in providing reversibility in energy transducing enzymes. And the implication is that proton-coupled electron transfer of a concerted nature is also very important in other types of electrocatalysis. Moving on, I mentioned earlier the vertical and horizontal rulers. Well, one of the ways that we can get back to more traditional enzyme kinetics is to take the view that the current that we're seeing is actually the rate at which the enzyme operates at a particular potential. And if we do things which will perturb that activity at a certain potential, we will observe the rate of change of rate. In other words, the rate of change of current. And I'm going to exemplify this in a few ways. This is the electrocatalytic voltammetry of an ordinary hydrogenase, which is obtained from E. coli, hydrogenase 2. I showed it a little bit earlier. This is the reversible region here. This is proton reduction. This is hydrogen oxidation. And at this point here, there's a bit of hysteresis because the active site gets over-oxidized and we go into an inactive state. Well, this inactive state, which is called nickel, which is called nickel B, or it's a nickel 3 hydroxide, was the subject of a lot of interest many years ago. And I'm not going to dwell on this very much, but the important point some 15 years ago was why is it that some hydrogenases cannot operate in oxygen, in air, or in fact are damaged by air, and others were quite easily able to operate in air. And the experiment that we carried out was to inject oxygen into the electrochemical cell, either under more reducing conditions where electrons are widely available, or under more oxidizing conditions. And we could do that under hydrogen, or we could do it under nitrogen. And then we can get rid of the oxygen, flush it out, and then trigger the step back to active enzyme with a potential step. So we'd be in a region here where the enzyme is largely inactivated, and we step the potential back. And what we see here is we obtain kinetics. We obtain the kinetics of the reactivation process. I'm not going to go any more into that. The important point is you can see processes very easily, which would be very difficult to see by any other method. The voltammograms I show here are for hydrogenase 1 and hydrogenase 2 produced by E. coli. I've mentioned hydrogenase 2 already. That would be represented by this gray trace here. Hydrogenase 1 is oxygen tolerant, and hydrogenase 1 is produced so that the organism can function under an aerobic lifestyle. It does not produce hydrogen. Certainly here you can see there's no hydrogen production, and there's a little onset potential. Now, we looked at the oxygen tolerance of this enzyme, and I can say that the oxygen tolerance of HYDE1 depends upon the ability to supply electrons to the active site when oxygen attacks, so that the product of the reaction is harmless water and not damage to the active site itself. Remember, the active site has got all these sulfur atoms in it which can be oxygenated. And in the next experiment, these were carried out by Non-Evans several years ago now. These show how a oxygen tolerant hydrogenase can be titrated with oxygen. This is here we've got 100% hydrogen. We drop it to 10% hydrogen at this particular point here, and then we add oxygen in little increments. We get a small drop in activity. We take the oxygen away. The activity returns. If we do a mutation on the enzyme where we've affected the iron sulfur cluster that's responsible for discharging an additional, not one, but 2 electrons back to the active site when oxygen attacks, what we see now is that the oxygen has a disastrous effect on that enzyme. And in fact, when we remove the oxygen at the end of the experiment, there's no recovery. Another example is looking at the binding of an inhibitor of carbon monoxide dehydrogenase. Thiocyanate, as one would expect, is because it resembles CO2, it's a competitive inhibitor of carbon dioxide reduction to CO. And here you can see experiments in which we've simply looked at the effect of adding this is actually, these experiments are actually adding this is actually yes, this is a mistake here. This should be an O atom here. This is cyanate. But anyway, what we're doing here is we're injecting cyanate, and we're seeing how fast the activity drops. We're using the current as our handle to determine the rate at which the enzyme becomes inactivated. This is actually reversible because we can wash the system out and get the activity returned. This is another example. This is an example where we're now using the electrochemistry to determine activation parameters. And what I show first of all is the active site of a nickel ion hydrogenase, in which we see just above the nickel and the iron atoms an arginine, which is suspending a guanidinium group over that active site. And we were interested in what this arginine group does in the catalytic cycle. Well, Non-Evans carried out quite a number of different mutations of this active site. In fact, she's really mutated every single residue that you can see in this little caption here. And what we found is if you change arginine to lysine, which conserves at least the formal positive charge, the rate drops to just about 1% of what it was before. But looking at voltammograms, of course, we're looking at current. And if we look at the current as a function of temperature, providing we've got a sufficiently high hydrogen concentration so that we're well above the Km for hydrogen, the temperature dependence gives us the enthalpies of activation of that reaction. And what I just show here, this is the enthalpy of activation of this variant R509K. You'd normally expect that slower reactivity, a slower reaction would be associated with a larger energy barrier, which is what we see when we replace the sulfurs by seleniums, put selenocystanes there. But in actual fact, the R509K variant has a lower activation enthalpy, and this tells us that the reaction now becomes entropy controlled, meaning that it was essential for this arginine group, the guanidinium group on arginine, to find itself in the right position in order that catalysis takes place. And so it's a probabilistic effect. In the next section, I'm just going to talk about what we call toys. And these are demonstrations that we managed to do using electrochemistry that have highlighted the particular facets of the enzymes as electrocatalysts. In the first case, it's possible to attach a hydrogenase or a carbon monoxide dehydrogenase to a semiconductor particle, so that if you excite electrons in the semiconductor and you're able to quench the holes formed in the valence band, the electrons that you send over into the enzyme will produce hydrogen for you, or reduce CO2 to CO. So this sort of work was first started several years ago by Erwin Reisner when he was in my group. And this is a very early example. We took anatase nanoparticles and we modified these with ruthenium BIPI photosensitizers, much like Michael Gretzel's work, attach a hydrogenase to them, and we could get very respectable hydrogen production rates from this. In a recent piece of work, Leon Zhang, who was a postdoc until 2019 in the group, she was interested in modifying hydrogenases so that we could put silver nanoclusters into the enzyme so that they would serve as part of the electron-transferring relay. Well, silver nanoclusters are themselves photoactive, and we found that when we could scaffold these hybrid enzymes onto anatase C3N4 heterojunction nanoparticles, we could get appreciable hydrogen activity. Now, this experiment is rather special because this is an oxygen-tolerant hydrogenase. And you remember that oxygen-tolerant hydrogenases don't normally produce hydrogen. And they don't do this because the distal cluster, the normal distal iron sulfur cluster, has a potential which is too high to allow the hydrogen to be produced. In other words, it acts as it acts to stall the electron. But by placing a silver nanocluster in the pathway of the electron, so that it now became the first site at which an electron was established in the enzyme, it's now possible to produce hydrogen. And moreover, we can do it in the presence of oxygen as well. In the final demonstration, we could power small electronics using very dilute hydrogen in air. And this is an example that we had in about 2015. We called it the hydrogen house. This was powered by a fuel cell, which has hydrogen and oxygen mixed together. And this was a little Christmas decoration that we had in the lab at that time. The point is about the uses of enzymes, purified enzymes in energy, is that it's very difficult to see that they would ever be useful in a real situation. The comparison is as I show it here. If you've got conventional electrocatalysts, they're stable, they're robust, they may have large overpotentials, particularly for the oxygen reduction reaction, but they are robust and they work. The comparison would be between a Land Rover and a Formula One car. because a Formula One car is very unstable. You have frequent pit stops. You have to change the wheels every so often. Part of the engine comes out and is replaced, but they're very fast. This is like the enzyme, and so they're inspirational. And the point therefore is that, of course, living organisms don't need to have highly robust electrocatalysts, because living organisms operate like pop-up shops. A catalyst is produced and used while there's a need for it, and it's then taken down and its components used for other purposes. So what would be a useful way of using electrocatalysis? Well, in the last part of my lecture, I'm going to talk about a platform that we discovered, which is called the electrochemical leaf, and it's a way of energizing enzyme cascades under rather peculiar conditions of nanoconfinement. Now, nanoconfinement has an important place in catalysis. We're probably all familiar with the way that zeolites would operate as catalysts. You confine reactants, and only the desired product is made and can escape. In living organisms, nanoconfinement is with us all the time. In a chloroplast, for example, or mitochondrion or other organelles, the enzymes are all packed together. Enzymes like operating in a production line, and of course these are not dispersed throughout the organism. They're highly localized so that the product from one catalyst is taken up as a reactant for the next catalyst, and so on. And in such a way, very high efficiency is achieved without escape of intermediates. And of course, the enzymes are present in very high local concentrations. How we do this is we buy a bottle of indium tin oxide nanoparticles, and then some of these are dispersed in dry acetone and iodine, and we apply a potential between two electrodes, one of which we're going to then use as our working electrode. And the nanoparticles condense together to form a porous conducting layer. And we can do this on various supports, such as graphite or titanium foil. And the important component here is this yellow enzyme, which you see in this vial here, FNR. What does it do? Well, it's going to permeate into this film, this layer, which is up to 3 microns thick on our electrode surface. Let's just look at FNR in a little more detail. It's a photosynthetic enzyme. It may be regarded as the mother of organic molecules because it takes electrons from photosystem 1 via a small carrier known as ferredoxin, and it converts these 2 electrons. There are two one-electron reactions which charge up the flavin center in FNR, and NADP is converted into NADPH. Nicotinamides are biology's hydride carriers. So 2 electrons and a proton equals a hydride, and many enzymes called dehydrogenases use NADPH or NADP as electron donors or electron acceptors, respectively. In the organism, the ferredoxin, which is represented here on the left in these 2 different views, forms a very tight complex with FNR, which is shown on the right side of each view, either as cartoon or a space-filling surface. And the electrostatic surface that you see here, predominantly blue for FNR and red for ferredoxin, tells us this is a strong electrostatic interaction. Well, FNR binds tightly in the hydrophilic and negatively charged environment of indium tin oxide nanopores. It binds at an incredibly high degree, and its binding increases as you increase the pH, which has the effect of making the ITO more negatively charged. At pH 9, if we count the charge under these peaks here, we have something like about 500 picomoles per square centimeter. This is about 100 monolayers. But of course, it's the high density is appearing because the FNR is dispersed into the pores. The electrochemistry is almost textbook. We have a cooperative 2 electrons reaction whereby the radical is partly stabilized. So you see from these half-height widths here, this is the background corrected, 57 and 59 millivolts are above the value for a strictly cooperative 2 electron reaction, which would be 45 millivolts, but below that for a 1 electron reaction, which would be 90 millivolts. So 2 electrons are able to bind with a fair degree of cooperativity to the FAD active site. And then this is the most important thing, that this catalyst, this FNR, which is now bound in the ITO pores, is able to recycle NADP and NADPH in a reaction that would conservatively be regarded as quasi-reversible, but is by far the most reversible and efficient electrocatalysis that anyone has achieved for nicotinamide cofactors. The result showed us that it should be possible to drive catalysis by dehydrogenases, particularly ones that are located close to FNR. And you see, simply by altering the potential here, you can oxidize or you can reduce nicotinamide in a very neat and efficient way. This next slide shows what we're actually aiming at here, because this is the network of ITO nanoparticles I represent in a cartoon form here, and we know that it's possible to put FNR in those pores, and other enzymes can be put into these pores as well. The result is it becomes possible to make extensive cascades, multi-enzyme reactions that occur because all the enzymes in the cascade are localized in the pores along with FNR. This is a reaction which would normally seem very unlikely from any electrochemist's point of view. It is the reaction of pyruvic acid and CO2 and ammonia via three further enzymes to make aspartic acid. So it's a synthesis of aspartic acid from pyruvate and CO2. And just focus on this voltammogram here. This green area here highlighted, this tells you it's reversible. And so we can go downstream from FNR to make aspartate. That would be going in the reducing direction, or we can go upstream to turn aspartate into pyruvic acid. That would be in this oxidizing direction here. And by rotating the electrode at high speed, we see that there are opposite effects for the upstream and downstream reactions. So picture this, really, the rotating disc electrode consists of an ITO layer on pyrolytic graphite, and into the pores of that ITO are placed FNR, malic enzyme, fumarase, and aspartate ammonia lyase, along with carbonic anhydrase, so that we can work from bicarbonate instead of CO2. And when we are doing the reaction in the downstream direction, the the current, this is negative current now. The current increases slightly when we increase the rotation rate and drops off when we decrease the rotation rate. And this is because we're speeding up the rate at which pyruvate can come into the pores. If we do the other direction, which is the upstream aspartate to pyruvate, the effect is opposite, because the effect of rotation is to increase the diffusion field, which in this case allows intermediates to escape slightly more readily. The enzymes don't escape by rotation. They're very firmly bound. And we come up with an interesting comparison with existing methods of studying nanoconfined cascades. Because what people do at the moment is they make a scaffold of in some way. It might be a moth or it might be DNA, and they put enzymes onto that, and then they look and see how, when the enzymes work together, the concentration of a product is produced at a greater rate in solution. Now, of course, we don't see the build-up of a product in the solution, not by electrochemistry at any event. We see the rate of reaction by current. In the last slide I showed you, we'd actually analyze the solutions after, so that's how we could determine the yields of aspartate and pyruvate. But in the cascade confined to an electrode, what we see instead is current, and we can evaluate very quickly how fast the reaction is going in any direction we choose by the value of the current that we actually see. And of course, we can drive the reaction backwards or forwards simply by changing the potential that we apply. Importantly, FNR is the transducer. It is the catalyst that drives the reaction in one way or another. And in the future, we expect that, first of all, about 1/6 of all enzymes are dehydrogenases. And it's very likely that this technology would allow extensive study of the properties of these enzymes. Furthermore, through building cascades that involve different enzymes, it may be possible to study a very wide variety of enzymes, even if they're not directly or involved in any redox reaction themselves, because all that we need to do is to see a current. Let's take this upstream direction. The current is produced because E2 will produce NADPH, which FNR will convert into electrons at the electrode. Of course, if we take out E3, which in our last example was a fumarase, this current would stop. So we have the opportunity to build artificial metabolons and establish how enzymes behave under nanoconfinement and very high concentrations. And we're looking at steady state kinetics of enzymes under very high concentrations. So it's rather different to the traditional world of enzymology. Well, that's really the end of my lecture. Rather than show a picture of the group, my group's quite small these days, what I show instead what I've put together, a Hall of Fame. And it does include some people who are still in the group and still in the ICL. You can pick out Claire McGarrity here and Non Evans. And further back, you can see Professor Vincent here. But all of these people, I've selected them because they have gone on to great things after being in the group, either as students or as postdocs. And I'm going to leave it there, and I'm very happy to answer any questions that you have. Thank you.
Thank you very much, Fraser. We do have one question already from Matthew. Thank you for the talk. I was wondering, what is the significance? Oh, it pops up on the screen. Love it.
Oh, right. Okay. What's the significance of Nicolines in some of these redox centers? What's the significance of nickel over iron? What you need here, because you've got a reaction where you've got to bind. If you're converting hydrogen to protons, so you need a metal that can bind a hydrogen molecule and then convert to a state in which it makes a metal hydride, because the proton has been taken away by a heterolytic process. So you need a metal ion that's going to stabilize a hydride, but at the same time, that metal has got to turn that hydride into a proton, and then the proton can escape. So of course, a hydride can't escape; a proton can. And so you need a catalyst that can span 3 oxidation states. So in this case, it would be nickel 3, nickel 2, nickel 1. In the case of iron, in the iron-iron hydrogenases, there's actually 2 ions there, and they share the load. What has always puzzled people in the field is that in the nickel-iron hydrogenases, the iron, which is an iron cyanocarbonyl compound, does not ever appear to change its oxidation state. All the redox chemistry seems to be happening at the nickel. So I don't know whether I've answered your question, but no other metals are known with hydrogenases. It's only either iron or nickel.
Great. If anyone has any other questions, feel free to put them in the chat. We'll give it a few minutes. And yeah. I guess one of my questions would be, what are the next steps of the lab in your research?
Well, I'm retiring, so so I hope to I hope that this particularly this new area that we're developing takes shape in other forms.
Well, since there aren't any other questions, thank you so much, Fraser, for giving a talk.
Okay, a pleasure. Thank you very much.
Thank you.